Hello everyone, this is Dr. Sonam and welcome to my channel Midha Physiology blog. So today we are on our fourth video related with the renal system. And before beginning today's objectives, let's revise briefly what had we discussed in the last video. So in last video we had basically discussed that how the blood is filtrated from these capillaries into the Bowman's space, and we had also come to know that this filtration process is also named as ultrafiltration. And when the blood had to pass through this capillary, we came to know that it had to pass through this endothelial and basement membrane, and then the podocyte food processes of the podocyte, which we together called the glomerular membrane, right? So all this thing had been discussed in the last video. So guys, if you want to know in detail about those things, you can check into my last video and the link is given in the description box. So let's begin with today's video, in which the objectives are to know. The objective is basically to know about the tubular part of the nephron. Okay, so till now we have finished about the initial dilated portion of the nephron, which we call the Bowman's capsule, and together with the glomerulus is called the renal corpuscle. So in this area the filtration of blood had occurred. Okay, so this much we had covered today. In up to now. Now today we are going to see that what would happen to the ultrafiltrate, or what is the course of that ultrafiltrate as it is passing down the nephron. Now we also know that these parts, okay, these all parts are basically the tubular part now, and this tubular part has been divided as the proximal convoluted tubule, and then loop of Henle, and then the distal convoluted tubule, and then the collecting duct, right? Now here in this slide I want to tell you one another thing, that in the capillary, in the glomerulus, you can see that these glomerulus, they are formed from the afferent arteriole. From the afferent arteriole, the network of the capillaries, they had been formed, and from the capillaries the blood was filtrated into the Bowman's space. And one of the peculiar feature of the renal system is that the capillaries, they are now draining into the arteriole, and this arteriole has been named the efferent arteriole, right? In other part of the body, the capillaries, they are basically draining into the venules, right? But here it is draining into the arterioles, and this arteriole is the efferent arteriole. And here what we have to now know is this efferent arteriole is again divided into the set of capillaries, okay, and these set of capillaries are basically going to give blood supply towards this tubular portion, this proximal convoluted tubule, or the loop of Henle, or the distal convoluted tubule. In all this tubule, okay, remember, when all this tubule are formed, inside there is a lumen in which the ultrafiltrate of the blood, they are passing, but surrounding the lumen in the wall there are the epithelial cells, and these epithelial cells obviously require the blood supply. So this blood supply are basically coming from the division of this efferent arteriole, and this division of the efferent arteriole, or when this efferent arteriole is dividing into the capillaries, they are forming two different kinds. So they can be the peritubular capillaries, or they can be the vasa recta. Okay, so these two type of capillaries can now supply different kind of nephrons. Now let us begin. Okay, now let us also see what is actually happening in the nephron. Okay, so this picture basically summarizes the whole renal system function. Here you can see that this whole structure is a nephron, okay, diagrammatic representation, in which you can see that in the Bowman's capsule, through the glomerular capillaries, the blood was being ultrafiltrated, and the ultrafiltrated product, the ultrafiltrate, right, it is now running through the lumen of the tubules. Now as it is being running through the tubules, lumen of the tubules, in this tubular portion, two kind of process can occur. So from the ultrafiltrate there can be reabsorption. So many of the product which has been filtrated from the blood, okay, those product, the water and the electrolytes, they are again being reabsorbed back into the circulation. Here you can see that these tubular portion are being supplied from the peritubular capillaries. So these peritubular capillaries, they are the division of the efferent arteriole, right? So this ultrafiltrate, the component of the ultrafiltrate, they can be reabsorbed back into the peritubular capillaries, and these peritubular capillaries, now they are draining into the renal vein, and this vein will now drain into the larger veins of our body and into the vena cava. In this way, again, these product, the ultrafiltrate, is now again being reabsorbed back into our body. Okay, so this is one process. The reabsorption can occur through the renal tubule or from the peritubular capillaries. Sometimes even the secretion can occur. Okay, some of the components, some of the ions can even be secreted back into the ultrafiltrate. So you see that from the first part of the nephron, the filtration had occurred, and then in the other part of the nephron, in the tubular part, two processes are occurring. The reabsorption is occurred, and then the secretion is occurring. Okay, and in this way, finally the urine constituent is being formed, and the waste product is being excreted out of our body. So these four processes, okay, this four process is basically the mechanism via which the urine is being produced in our body. Firstly, there should be filtration, and then reabsorption, and then secretion, and then there will be excretion through the urine. So this filtration portion, it is occurring in the glomerulus, in the Bowman's capsule, right? And the reabsorption and the secretion process, it is now occurring in the renal tubule. To know how this reabsorption and the secretion process is occurring in the renal tubule, firstly we have to know how this renal tubule, what are the parts of the renal tubule, and what its structure. Okay, when you come to know about the structure, then only the understanding of the function would become easier. So today we are going to briefly learn how these various kind of tubules look like. Okay, so firstly we'll start with the proximal convoluted tubule. So this proximal convoluted tubule, you can see this proximal convoluted tubule is the first part of the renal tubule, and it is close proximity to the Bowman's capsule, right? And it is coiled in nature. So obviously the Bowman's space is in continuous with the lumen of the proximal convoluted tubule, right? So here you see it is continuous with the with that of the Bowman's capsule space. Okay, and it is 15 millimeter long. So here let us see how does the cell look like in the proximal convoluted tubule. Okay, so these are the cells, you see. So these cells which is forming the lining of the proximal convoluted tubule, the cells are cuboidal in nature. This epithelium, they are made up of cuboidal cells. Okay, now another important thing is how you are going to read the slides, read the diagrams when you are going to study the renal system. Okay, so whenever you are going to understand the picture, firstly know that which one the epithelium they are facing to which side. Okay, so this epithelium, okay, this epithelium, in one side there is the tubular lumen. That means in this portion the ultrafiltrate it is coming. So this portion of this epithelium which is facing towards the tubular lumen, you call this portion as the apical portion. Okay, and the opposite portion, this opposite portion or the opposite side which is lying in the basement membrane, you call it basal side. Okay, so I again repeat, facing towards the luminal side, this side of the epithelium, it is the apical side, and this one is the basal side, right? And in the basal side, towards the basal side, you can see the peritubular capillaries, they are even giving the blood supply. Okay, so here this particular diagram is the epithelium of the proximal convoluted tubule, and as I had told you that it is a cuboidal epithelial tissues. Okay, and in the apical portion you can see that there are many finger-like projection which you call microvilli. Okay, so these are the microvilli which is present in the apical side of the epithelial lining cells of the proximal convoluted tubule. Okay, now as there are so many microvilli, you can see that because of so many folding, the total surface area in the apical side it is increased. Okay, and it is very, very important mechanism. It is very important concept that due to increase in the total surface area in the apical side, many reabsorption process it basically can occur through this apical side present in the proximal convoluted tubule. Later on, you'll come to know that when the ultrafiltrate is being formed, it is carrying water, the electrolytes. Okay, and when we are talking about electrolytes, mostly sodium, potassium, glucose, they are very, very important, right? And most of these electrolytes they are being reabsorbed into our body by the proximal convoluted tubule only, and that reabsorption process is becoming easier because the total surface area in the apical side it is larger. Okay, Okay, now the another feature which I want to show you over here is you can even see that in the apical side the two epithelial cells, they are connected with each other with the tight junction. Okay, so this tight junction is basically forming a barrier to prevent the leakiness of the fluid and other substances in both direction, from this tubular side into this side, or from this side into the tubular side. Okay, luminal side. So it is just preventing the leakiness of the fluid in either side, and this tight junction is not being seen in the basal side of this epithelial tissue, and in between there is a space which you call lateral intracellular space. Okay, so this feature you have to understand when we are talking about the proximal convoluted tubule. Okay, so again want to show this process of reabsorption occurring in the proximal convoluted tubule. The picture, the diagram is not so clear, but it will give you idea. Okay, so here again this is the luminal side in which the tubular fluid, that is the ultrafiltrate, is running. So obviously this is the apical membrane or the apical side. Okay, and this one is the plasma running. So this is the capillary. So this is the peritubular capillary which is present in the just at the basal side of the epithelial tissue. Okay, so here in this apical membrane of this epithelium of the proximal convoluted tubule, there are many microvilli which is increasing the total surface area, and in this microvilli you can see they are the transporter. Okay, so this transporter are now transporting the electrolytes which is present in the tubular fluid or in the ultrafiltrate into the epithelial cell, and from the epithelial cells now it is being transported out. Okay, they are transported outside, and from where it is now being transported into the capillary. So in this way the reabsorption is occurring. Okay, there should be the presence of transporter in the either side, then only it is being transported, the substances are being transported into the capillary and then reabsorbed or circulated back to our body. Now the things are being repeated in the slide again. So you can see that the cells in the cells forming the epithelial lining in the proximal convoluted tubule, they have numerous microvilli which is increasing the surface area at the apical or luminal side, and in the tubular side there is also connected, the cells are also connected at the apex by tight junction, whereas in the basal side there is the extension of the extracellular space which you call lateral intracellular spaces. Okay, so later on when we are going to study about the reabsorption process in detail, this feature should be in your mind. Now this proximal convoluted tubule is again been divided into two parts, the pars convoluta. Okay, that is the initial portion in which the proximal convoluted tubule or the proximal tubule, they are convoluted or they are coiled. Okay, and it is followed by the straight portion of the proximal convoluted tubule which you call pars recta. Now the other features which you can see in the proximal convoluted tubule are these cells. Okay, the epithelial cells, they are rich in mitochondria, obviously because most of the reabsorption they are going on through this proximal convoluted side only, the epithelium only. So obviously they require more energy and there will be more mitochondria, and they would also consume more oxygen. Okay, their oxygen consumption would be higher, and then you can see that there is the maximum absorption. It is not absorption when we are talking about the renal system, it is reabsorption. Okay, so there is the maximum reabsorption of the electrolytes from the PCT or the proximal convoluted tubule like sodium. So these things should be clear in your mind now. The second part, okay, from the proximal convoluted tubule, so the ultrafiltrate is now running in the proximal convoluted tubule, and then in the straight portion of the proximal tubule which we call pars recta, now it is running down, it is descending down through this tubular portion and again ascending up. Okay, and while descending and ascending up, it is forming a loop-like structure, and which we call the loop of Henle, discovered after the Henle name given after the scientist who has discovered it. Okay, now this loop of Henle, obviously as it is descending down and ascending up, it is carrying two portions, descending limb and ascending limb. Okay, and you can see that the descending limb is thinner than the ascending limb. Okay, so ascending limb is thicker, however the initial portion of the ascending limb is also thinner. So okay, the thinner loop of Henle, okay, the thinner loop of Henle means the thinner descending limb as well as the thinner ascending limb. Okay, so this thinner portion of the loop of Henle, its length is around 2 to 14 millimeter. Why 2 to 14 millimeter? Because sometime this thinner ascending limb of loop of Henle may be absent. Okay, in which case the thinner loop of Henle will be shorter, and if it is the thinner ascending limb of loop of Henle, if it is present, is the thinner segment whole its length would be increased, and this thinner segment is lined by the flat squamous epithelium, and then after the thinner portion, the ultrafiltrate will now be draining into the thicker ascending limb of loop of Henle, and its length is around 12 millimeter, and it is made up of low cuboidal epithelium. So here you can see that only the thinner portion of the loop of Henle, its size, its length is being variable, okay, from 2 to 14 millimeter. And this portion, okay, this loop of Henle, the thinner portion, if is only 2 millimeter, the whole length of the nephron itself would be shorter, and if it is longer, the thinner loop of Henle, it is longer, that is 14 millimeter, the whole length of the nephron would be longer, okay. So this short loop of Henle, in which the ascending thinner portion of loop of Henle is absent, okay, such kind of shorter loop of Henle and eventually the shorter nephron, this kind of nephron, they are present in the cortex of the kidney. So this kind of nephron, you call it cortical nephron, whereas this longer loop of Henle, okay, the nephron with the longer loop of Henle, they are present nearby the medulla. So this kind of nephron, you call it juxtamedullary nephron, okay. So accordingly, now we can see that the nephrons, they are of two types, the cortical nephron and the juxtamedullary nephron, okay. I again repeat, why this loop of Henle, it is shorter, because the thinner ascending limb of loop of Henle, it is missing, okay. And if there is thinner ascending limb of loop of Henle as well as thinner descending limb of loop of Henle, the loop of Henle itself, the length would be longer, okay. Now let us see what are the cortical and juxtamedullary nephron. Remember the differentiating feature between the cortical and juxtamedullary nephron, it is very much important from the examination point of view. So here the first diagram, it is the cortical nephron, and the second one is the juxtamedullary nephron, okay. So whenever we are talking about this term juxta, remember that juxta means near to, okay. So here juxtamedullary means near to the medulla. So here the nephrons, they are lying near to the medulla, so you are calling it juxtamedullary nephron. Here, okay, let us see from the cortical nephron. So the cortical nephron, its Bowman's capsule and its most of the part, it has been initiated in the renal cortex only. Here you can see that this lighter pink color portion is the renal cortex and the darker one is the renal medulla. So in case of cortical nephron, this nephron has started from the cortex only, and then there is the loop of Henle which is shorter, and then it is draining, it is followed by the distal tubule and then into the collecting duct. Now if you are comparing this nephron with this juxtamedullary nephron, you can see that the whole length of the juxtamedullary nephron, the nephron itself is so long, because you see that the loop of Henle in the juxtamedullary nephron, it is so longer in comparison to the cortical nephron, right? So here you can see this juxtamedullary nephron, it has started just near to the medulla portion, right? And the loop of Henle, it is so longer, due to which the length of the nephron itself has become so longer, okay. Now the another differentiating feature or the important features which you have to know about this nephron is, when we are talking about the kidney, in our kidney, when we are talking that the microscopic structure is nephron, now we came to know that the nephron are of two types, the cortical nephron and the juxtamedullary nephron. Remember that in between these two nephron, the cortical nephron or the nephrons which is found in the renal cortex, they are maximum in number, okay. So 85 to 86% of the nephron which is present in our kidney, they are cortical nephron, and they have the small glomeruli, and obviously there is short loop of Henle, and the shorter loop of Henle is because there is no thinner segment of ascending loop of Henle, okay. And these cortical nephrons, they are being supplied by the peritubular capillaries. These peritubular capillaries, they are the division of the efferent arteriole. So here these peritubular capillaries are supplying the cortical nephron, and the rate of filtration in the cortical nephron, it is slower. Okay, remember, the rate of filtration in the cortical nephron, it is slower, due to which slowly and carefully the unwanted waste product of the blood, they are being excreted. Okay, so the main role of the cortical nephron is it is associated with the excretion of the waste product in the urine. So these are the features which you can see in the cortical nephron. Okay, now another important nephron is the juxtamedullary nephron. This kind of nephron, they are fewer in number. Okay, the percentage is only 14 to 15%. That means in our kidney, only 14 to 15% they are the juxtamedullary nephron. They have the larger glomeruli, and where are they located? They are located between the cortex and medulla, or nearby the medulla. And they are the nephron itself is so long because their loop of Henle, it is longer. Why the loop of Henle it is longer? Because the thin segment contain both descending and ascending limb of loop of Henle. Okay. Now, while talking about its blood supply, let us see about this nephron, juxtamedullary nephron. Since it is so longer, now not only the peritubular capillaries is sufficient to supply it, but there is again another division of the efferent arteriole, that is vasa recta, is also now going to supply this kind of nephron because it is so longer. Okay, so here I again want to repeat that the cortical nephron, it is many in number, which is found in the cortex. The juxtamedullary nephron, it is only 15 to 16%, and it is found nearby the medulla. Okay. Now, sometimes in the MCQ, they may ask you that where the renal blood supply is higher. Is it in the renal cortex or is it in the renal medulla? Here, by knowing the percentage of this the nephron only, you can estimate that the blood supply should be higher in which area. Okay. Like, we came to know that the nephron in the cortex, they are in so larger percentage. That means obviously the blood supply in the cortex should be higher in comparison to the blood supply to the renal medulla. Okay. Now, when you are talking about the juxtamedullary nephron, we came to know that it is not only being supplied by the peritubular capillaries, but also the vasa recta, they are going to supply it, and the rate of filtration in the juxtamedullary nephron, it is higher. Okay, and this juxtamedullary nephron is basically associated in the urine concentration as required, or it is associated with the counter current system or counter current mechanism. Okay, so here this counter current mechanism is itself a very vast topic and important concept in the renal physiology. The basic idea which the counter current mechanism is trying to tell us is, like we see in our daily life also, when we are consuming lot of water, then at that time we have to go and urinate frequently. But if we are consuming lesser amount of water, the frequency of urination is also decreased, right? So taking larger quantity of water or lesser quantity of water is not a pathological condition. It may be our different kind of daily routines, right? So in normal condition also, we came to know that the kidney has the capability of either concentrating urine or diluting urine according to the hydration status of an individual. Okay, so that mechanism in which the urine is being concentrated or diluted as per the requirement or as per the hydration of the individual, you call it counter current mechanism. Okay, so this urinary concentration function, whether the urine has to be produced in a diluted form or in the concentrated form, this function is being done by the juxtamedullary nephron. So these are the differentiating feature between the cortical nephron and the juxtamedullary nephron, and remember these differentiating features are very, very important. So okay, now we are talking about this loop of Henle, right? We had come up to this thicker ascending limb of loop of Henle. Now from the from the PCT, firstly from the glomerular capsule, the blood was ultrafiltrated, going into the PCT, going into the loop of Henle. The last portion of the loop of Henle is thicker ascending limb of loop of Henle. Okay, now here another interesting feature is, when the tubule portion is forming into this thicker ascending limb of loop of Henle, and when it is now followed by the distal convoluted tubule, here you can see this portion now, okay, this tubular portion now is coming again back to the renal corpuscle. Okay, when we are talking about renal corpuscle, we had told that from this area the tubule has arisen, so this is the tubular side, and from this area the vessels, they are invaginating, so this is the vascular side. Now in this vascular side only, okay, resting between the afferent arteriole and efferent arteriole, now the thicker ascending limb of loop of Henle is being followed by the distal convoluted tubule. Okay, so here at this area, when this thicker ascending loop of Henle is being followed by the distal convoluted tubule, the cells which were the cuboidal in nature, these cuboidal cells are now being differentiated or modified into the columnar cells. Okay, this modified columnar cells, we call it macula densa. Now what is this macula densa and what is function? This thing we'll discuss in the juxtaglomerular apparatus, which I'll cover in a separate video. Okay, so again, you can see that the thicker loop of Henle, when it reaches the glomerulus of the nephron, nestles between the afferent and efferent arteriole, and the cells become modified into the columnar cells to form the macula densa, which are the part of the juxtaglomerular apparatus. So till now, at least you have to understand this much point. Okay, now we'll see the distal convoluted tubule. It starts at the macula densa. So what is this macula densa? These are the modified cells which is present at the end of the thicker ascending limb of loop of Henle. Okay, so this descending convoluted tubule, it is 5 mm long, and it is characterized by the low cuboidal epithelium with fewer scattered microvilli. Okay, so whenever there is microvilli, obviously it is associated with increasing in the surface area so that the reabsorption can occur. Now let us see this collecting duct. Now this distal tubule is followed by the collecting tubules or the collecting duct. Okay, now these collecting tubules, they are 20 mm long. Okay, and they are made up of this clear cuboidal epithelium, and in the collecting duct, now the ultrafiltrate is passing, and through which the many other reabsorption process in this collecting duct, also the other reabsorption process can also occur, and then the final constituent of urine is being formed, and it will drain into the renal pelvis and into the ureter. Okay, now let us see what is happening in this distal, in this collecting tubule. So here you can see two different kind of cells in the collecting tubule. Okay, in this picture only you can see. Okay, so here you can see, as the ultrafiltrate is coming from the distal tubule into the collecting duct, the water is being reabsorbed. Okay, so when we are talking about the reabsorption of the water from the renal tubule, from our previous knowledge in the physiology only, we came to know that it is because of the antidiuretic hormone. The posterior pituitary is releasing this antidiuretic hormone, and this antidiuretic hormone is acting in the renal tubule for the reabsorption of the water. So this thing we had learned, right? So where is this reabsorption of the water occurring? It is occurring in the collecting duct. So this is one of the very principal function of the collecting duct, that on the presence of the antidiuretic hormone, the water reabsorption would be increased in the collecting duct. Okay, so how this is occurring, you can see that from the plasma in the peritubular capillary, okay, the antidiuretic hormone which was secreted from the posterior pituitary, it is coming, and it is now acting over the receptor which is present in the basal side, of this epithelium of the collecting tubule. This receptor is the V2 receptor. Okay, the V2 receptor is the G protein coupled receptor. Now via the GS subunit, it is going to cause the activation of the adenylyl cyclase, and there will be increase in the cyclic AMP, and eventually they are going to cause the insertion of this aquaporine channel into the apical membrane. Okay, so in the tubular fluid or in the ultrafiltrate, whatever water was coming, now through this aquaporine, which is a water channel, through this channel, now the water will be reabsorbed into the epithelial cells, and from the epithelial cells back into the capillary, and from the peritubular capillary, now it will be reabsorbed back into our body. So in this way, the primary function of the collecting tubule is to reabsorb the water, and this principal function is being done by the principal cell. Okay, so in this collecting tubule also there are two type of cells, the principal cells which is taller, and its main function is under the effect of the antidiuretic hormone or the vasopressin, it is causing the water reabsorption, and it is also associated with the sodium reabsorption. And apart from the principal cell, there is also intercalated cells, and the main function of the intercalated cell is associated with the acid or bicarbonate transport. Okay, so this thing you can see in this diagram. You can see here, this is the principal cell. In the principal cell, you can see this is the luminal side and this is the blood side. Okay, so whatever thing is being reabsorbed from the ultrafiltrate coming into the lumen, now it will be reabsorbed into the blood, right? So here in the ultrafiltrate, if the sodium is coming in the principal cell, in the apical side, there is the epithelial sodium channel from which the sodium would be reabsorbed, and this reabsorbed sodium is now being given back to our circulation. This is again another function of principal cell apart from the reabsorption of water. And these two cells, okay, the IC, these are the intercalated cell. So this one is the A intercalated cell or the acid intercalated cell, and the B is the associated with the bicarbonate intercalated cell. Okay, so in this intercalated cell, you can see that in the apical side, because of this transporter, the hydrogen ion is being secreted outside. Okay, similarly in the B intercalated cell, you can see that the bicarbonate, okay, in the later portion you can see the bicarbonate, it is being reabsorbed back. Okay, so this intercalated cell is basically associated with the acid base transport, so as to maintain the hydrogen ion concentration in the ultrafiltrate of the lumen. Okay, so these were the functions of the distal convoluted tubule. So I again revise that I have told you all this thing in very detail, because when we would be discussing about the reabsorption process which is occurring in the nephron, if you understand this much anatomy or this much portion in detail, this much basic thing in detail, understanding those video will be very much easier for you. Okay, so let us revise what have we learned in today's video. So we had started with the PCT, right? The PCT also had two portions, the convoluted and recta, the pars recta, and then we came to know about the loop of Henle with the thinner and thicker segment. Accordingly, we had divided the nephron into the cortical and the juxtamedullary nephron. And I again repeat, the differentiating feature between the cortical and juxtamedullary nephron are very, very important to understand the renal physiology. And then after that, we came to know that at the end of the thicker ascending limb of loop of Henle, the cells were modified into the macula densa cell. Okay, so what is this macula densa cell? I will subsequently cover in next video. And after this thicker ascending limb of loop of Henle, it is followed by the distal convoluted tubule and then into the collecting duct. In the collecting duct or the collecting tubule, we also came to know that there are two kinds of cells, the principal cells and the intercalated cells. And we came to know that how antidiuretic hormone is acting for the reabsorption of the water, and how even the sodium can be reabsorbed back from the collecting tubule. Okay, so this much is what we at least require to understand the reabsorption process now, and this much for today also, guys. So if you guys like this video and is helpful to you, please like, share and subscribe to our channel. And I again say that if you this video was also a little bit longer, but understanding these points are very, very essential. I again repeat, very, very essential. And if you understand this basic feature only, then you can understand the reabsorption and other phenomenon going on in the nephron. Okay, so yes, so if this video was useful, please like, share, subscribe, and yes, please do comment to our video. If you have any kind of queries and suggestion, please do comment, because those things are very much helpful to me to make better kind of videos in future. Okay, so thank you guys.